All right, tough act to follow. This is not a clinical talk. This is a hypothesis-generating basic science type talk. So my name is Peter Bajic from the Cleveland Clinic, and I'm going to be talking about penile prosthesis biofilms, friend or foe. So my disclosures, I'm a consultant for Boston Scientific and Coloplast. I have research funding from Coloplast, and what I'm presenting here is research supported by the 2020 SMSNA Young Clinicians Research Grant, and also some funding from Coloplast. So we know that penile prosthesis infection is a rare and devastating complication. It affects up to 5% of virgin cases, up to 13% of revision cases, more common in diabetics, revision cases, and severe fibrosis. Treatment is removal with or without replacement, and the historical dogma that we all know is that antibiotics cannot cure a true implant infection due to biofilm on the device that prevents you from being able to eradicate that infection. So many infection prevention measures have been attempted or studied. Antibiotic coatings, hydrophilic implants, perioperative antibiotics and irrigations, alcohol-based prep, no-touch techniques, negative air pressure, making a shorter surgery. All of these things are important considerations for implanters. But is a sterile implant really achievable? Conventional culture data, so the types of cultures we obtain in clinical practice, show that 25% of implant spaces have bacteria even after antiseptic washout. That's dating back to the 2000s with some work from Gerard Henry. And 70% of implants with detectable bacteria and biofilms do not have symptoms of clinical infection. This has more recently been corroborated by data using DNA sequencing that show that 93 to 100% of explanted devices, whether they're infected or not, have detectable microbes on them. So this is some scanning electron microscopy imaging from our recent publication. So if you look at the top three panels there, these are sterile devices straight out of the package. They do not show any biofilm. We have a piece of the pump, cylinder, and rear tip extender. And then the other images just below those are the same device components, but from a device that was removed from a patient with mechanical failure that was not infected. And you can clearly see biofilm on all components. So really, sterility in the revision setting is impossible. This is the tissue capsule around the implant that you can also see biofilm on. So if we take a step back and just look at the human body, we're comprised of 98% human and 2% or 3 pounds of bacteria. If you look at the number of cells, it's 90% bacterial cells and 10% human cells. And if you look at the DNA, over 99% of the DNA in the human body is actually bacterial DNA, and this comes from microbiome research. So what is the microbiome? It's essentially an aggregator, a community of microbes, not just bacteria, but viruses and all sorts of organisms that live in a particular environment, along with the genetic material of those devices, which is what we study. The Human Microbiome Project identified, based on these communities of microbes all around the body, associations with various diseases, everything from inflammatory bowel disease to kidney disease, et cetera. And really, the microbiome is very familiar to urologists. We know that bacteria in the body affect stone pathophysiology. Bacteria in the vagina can be related to UTI risk. Lactobacillus can prevent E. coli adhesion. And we've actually used bacteria, Mycobacterium bovis, as a treatment for bladder cancer. That's what BCG is. So really, in that case, we're manipulating the microbiome of the bladder. So good bacteria can keep bad bacteria at bay by competing for resources. So if we have a healthy community of bacteria, like in our mouth, we don't have infection, same thing in our gut. So a microbiome does not mean infection. So how do we study these communities of microbes in various parts of the body? We can use metagenomics, which tells us which types of microbes and genes are present. We can use metabolomics to see what those microbes actually do. And then this can potentially lead to changes in how we diagnose and treat various conditions. So the first publication from our group in 2023 looked at 27 explanted penile prostheses, artificial urinary sphincters, a couple other devices. Most were non-infected, and 93% of them had biofilms. So we used bacterial gene sequencing and identified Staphylococcus, Escherichia, which is what E. coli family is. They were both abundant in both infected and non-infected devices, but we also found some less common and unusual bacteria like those listed here that were more prevalent in infections. Interestingly, over a third of devices identified had rifampin resistance genes, and there were many other resistance genes that were identified. I just want to turn your attention to this image on the right. So this is looking at diversity of the communities on the surfaces of these devices. So when we talk about diversity, it's like how many different types of microbes are present. So devices that were implanted for longer prior to removal actually had more diversity in their communities. So a device that's been in the body for 10 years has more different types of microbes on it than one that's been implanted for 1 year. When we looked in this study at differences between non-infected and infected devices, there was no difference in diversity of those communities between infection and non-infection. Same thing with diabetes. This study also interestingly looked at how the microbes and their metabolites, or the substances that they produce, interact. And we used what's called micrometabolite networks that showed there's really these complex interactions that happen. It's not like a single pathogen that necessarily drives that transition from a colonized state to an infected state, but it's all the interactions between the different microbes, their different products, including the biofilms that they produce. So there were differences between what infected communities looked like and non-infected communities. A subsequent study published in the Journal of Sexual Medicine from our group looked at 20 IPPs, 17 were non-infected, 3 were infected. We took them out prior to administering any antibiotic irrigation. They did receive intravenous antibiotics, but basically we subjected these devices that were removed to sonication to dislodge, ultrasound basically, to dislodge the biofilms and bacteria that were on them, and then used DNA sequence, or 16S, which is bacterial RNA sequencing, shotgun sequencing, and scanning electron microscopy to look at the presence of biofilms, microbial diversity, what types of bacteria, viruses, et cetera, were present, what types of antibiotic resistance genes were present on the different components of each device. So we found that microbial diversity, so how diverse those communities were, varied. It was different for each patient, and there were some differences based on the device manufacturer, meaning one manufacturer versus the other. But interestingly, within a single patient, there was no difference between the different components. So what was seen on one component was very similar to what was on the different component of the same device. Interestingly, there were no differences depending on whether or not the patient was infected or not. We did find significant differences in bacteria, viruses, and metabolic pathways between the different device types, and we consistently found antibiotic resistance genes and biofilm-associated genes. So this is 20 individual patients. This is a lot of pretty colors here, but what this means, each vertical bar is an individual patient, and the different colors are different types of bacteria that were present. So just to demonstrate to you, a lot of colors means a lot of different bacteria identified. Not all of these are pathogens. Most of these are non-pathogenic bacteria. And then when we look at, let's say, an individual patient, the different components were very similar. So the colors that you see here, although the sizes of each color are a little bit different, same colors depending on if you're looking at the capsule, cylinder, pump, or reservoir. So in this study, we looked at the types of bacteria, comparing the 2 different manufacturers of these devices, and we anonymized them to A and B so that nobody would try to draw conclusions about infection. This is not the type of data that you can make some sort of conclusion about infection. That's not what we're looking at. But there were also differences, not just in bacteria, but also in viruses. When we looked at the different pathways, metabolic pathways, different metabolites that they were producing, there were also differences between the 2 manufacturers. So if we look specifically at what those things were, on the left there, those are the bacteria that were different between one, or more enriched in one manufacturer's devices versus the other. On the bottom there, a little bit cut off, but those are the metabolic pathways that were different, and then on the right there, viruses. So this does not mean that those are, bacteria that lead to infection. I cannot emphasize that enough. But what we're trying to get to the bottom of is how these different microbes interact with one another, and what might take something from an equilibrium state where there's no infection, just like in your gut, just like in your mouth, to becoming an infection that actually leads to inflammation and clinical sequelae that ultimately leads to removal. So what have we learned from the penile prosthesis microbiome? Biofilms are ubiquitous, even in the absence of clinical infection. Complex interactions between the microbes and their metabolites drive the infection process. Device and patient factors are important and influence biofilm composition. There's lots of antibiotic resistance genes, but we don't fully understand what the significance of those is, and their role is complex. And we need to kind of change our mindset from focusing on, eradicating biofilms completely, because it's not feasible, to trying to better understand how to set up an environment of a healthy, protective biofilm that does not lead to infection. So what's next? We want to better understand how do those microbes interact with one another, the biofilms and the host, meaning the person, and what triggers that conversion from a stable, symbiotic community to a what we call dysbiotic or infected state. So these are preliminary findings that set the groundwork for a more nuanced, personalized approach to treating and preventing infections in prosthetic urology. Thank you. Oh, last thing. So if anybody wants to read a cool book, I always recommend this one, just a quote from this book, which is about microbiomes. Every one of us is a zoo in our own right, a colony enclosed within a single body, a multi-species collective, an entire world. Thank you.